Hi, this is Dr. Edelberg, and welcome to Biology Video Notes number 4. This video is on controlled experiments. Whenever possible, a controlled experiment should be utilized. However, it may not be always possible to utilize a controlled experiment. For example, an environmental scientist working out in the environment would have an impossible task trying to control for every factor in the environment. So what is a controlled experiment? A controlled experiment is an experiment where you run two or more experiments side by side and keep every factor or variable the same except for one. For example, let's take the question, does fertilizer increase plant growth? Well, before we design our controlled experiment, let's make an if and then statement. Well, what is an if and then statement? After the if, you put your hypothesis. After the and, you put down your test. And after then, you put down a prediction of your results. So, for example, with the question, does fertilizer increase plant growth? We could say, if fertilizer increases plant growth, and plants with fertilizer are compared to plants without fertilizer, then plants with fertilizer will grow more. So note, if fertilizer increases plant growth, that's our hypothesis. That's what we think the answer to the question is. And plants with fertilizer are compared to plants without fertilizer, that's our test. We're growing plants with fertilizer, and we're comparing them to plants that we grow without fertilizer. Then plants with fertilizer have increased growth. That's a prediction. We're saying, well, fertilizer increases plant growth, and we compare them to plants without fertilizer, we're predicting that they should grow more. So that is an if and then statement. All right. So in this case, what do? We set up two groups of plants. One group of plants has fertilizer, one group of plants does not have fertilizer. We said that everything is kept the same between the two groups except for the fact that one group has fertilizer. Right? The group that has the factor that's being tested or being changed is the experimental group. The group that lacks this factor is the control group. So in our case, the experimental group is the group of plants with fertilizer, and the control group is the group of plants without fertilizer. All the things that are being kept the same are called constants or control variables. So that could include type of soil, the amount of soil, the amount of light, amount of water, when the water is given, the temperature, etc. All those things have to be the same between the two groups. Those are your constants, those are your control variables. Another variable is the independent variable. The independent variable is the one thing that is different between the control group and the experimental group. We could also say that the independent variable is what the scientist is changing or what the scientist is manipulating. The other variable is the dependent variable. The dependent variable is what's being measured. Another way of defining the dependent variable is that's the variable that's responding to the independent variable. So if the independent variable in our case is fertilizer, then how does the plant respond? It responds by growing more. So the dependent variable, again, is what we're measuring. So we can say that the dependent variable is the height of the plant. One could also have several levels of independent variable. So for example, we could set up 3 or 4 experimental groups. We could have one experimental group where all the plants are given 5 milliliters of fertilizer. We could set up another experimental group that's given 10 milliliters of fertilizer. And we could set up a third group of plants that are given 15 milliliters of fertilizer. So the independent variable is the same in the sense that the only thing we're changing is fertilizer. It's fertilizer in every single case, but we're giving different amounts of fertilizer. So this is known as different levels of the independent variable. Again, note that we're still only changing one factor. One group lacks fertilizer, the other group contains fertilizer. All right, so what's the importance of a control and a controlled experiment? Well, there are really 3 reasons. First, it helps make better judgments about results. For example, if we just grew plants in fertilizer and they grew, 200 feet, can we really say that fertilizer increased plant growth? No, because we have nothing to compare it to. Perhaps the plants without fertilizer would grow 300 feet tall, and then our conclusion would be that fertilizer decreases plant growth. Of course, if the plants without fertilizer grew 10 feet tall, then we could say absolutely fertilizer helps increase plant growth. And then the other possible answer is that the plants without fertilizer grew just as tall as the plants with fertilizer. So then we can make the conclusion that fertilizer makes no difference. But without a control, we cannot make any of those judgments or conclusions. And the second reason, it helps us to know what factor is responsible for the change. If we didn't control the experiment, if each group had different temperatures, different amounts of sunlight, fertilizer, no fertilizer, how do we know that fertilizer was responsible for the change and not the difference in sunlight or the difference in temperature? But if all those things are kept the same and the only thing is different is the fertilizer, then it must be the fertilizer that's causing the change. So again, it helps us know exactly which factor is responsible for the change. All right? And lastly, it helps us figure out or pinpoint what went wrong if something goes wrong with the experiment, if a mistake is made or it just doesn't go the way it should go. And a great example of that is that demonstration that we did in class with the iodine and the potato. Remember, we put the iodine on the potato, and we expected the potato to turn black because iodine turns black in the presence of starch. And we were shocked. The potato did not turn black. The problem with that experiment was we didn't have a control. If we had a control and we had a known solution of starch, which the second time around we did, right? So we took this known solution of starch, we put the iodine in there as the control, and then we put the iodine on the potato, and what did we notice? The known starch didn't turn black, and the potato didn't turn black. So we knew that there was something wrong with the iodine. So what did we do? We got a new batch of iodine, we redid the control experiment, put that new iodine into the starch, it turned black. We knew this iodine was good, and when we put it on the potato also turned black. So thanks to a control, we were able to pinpoint a mistake or figure out where the experiment was not behaving as should. Okay, let's check for understanding. Let's use as an example for a good test question the following. Does antibiotic X kill staph bacteria? Number one, come up with a good if and then statement. I'll give you a moment to think about it. So you want to think of what your hypothesis is and put that as the if. What your experiment is, put that as your and then what you predict the outcome will be and use the then. Right, you think you have it? Alrighty, well, if it goes something like this, then you did a good job. If antibiotic X kills staph bacteria, and the staph bacteria are placed on plates with antibiotic X and plants without antibiotic X, then there will be less staph bacteria on plates with antibiotic X. So hopefully you came close or did very well. Next question. What would the independent variable be? Well, think about it. What's going to be different between the two groups? Right, antibiotic X. So antibiotic X would be the independent variable. I'm sure you can guess the next question. What is the dependent variable? So what changes the number or amount of bacteria? All right, good job. What might be some constants? Well, the size of the plate, the amount of food given the bacteria, the type of bacteria, the amount of water, the amount of light, the temperature. These are all other things that you might want to keep the same. Not might, but definitely want to keep the same between the two groups. What might be different levels of the independent variable? You think you have it? Alrighty. Well, we can use different amounts of the antibiotic X. We could use 1 milliliter, 2 milliliters, 3 milliliters, 4 milliliters, and test them all together against the control. All right, very good. Thank you.